Добрый вечер, уважаемые слушатели. Сегодня мы открываем наш ежегодный курс лекций, который состоит из 3 лекций и подготовлен при поддержке магистерской программы «Когнитивные науки и технологии от нейрона к познанию» НИУФШи. И сегодняшняя лекция пройдет на английском языке, поэтому я переключаюсь на английский. Hello everyone, thank you for coming. Today we are opening our annual lecture course, which was prepared with support of master's program called Cognitive Science and Technologies from Neuro to Cognition. And now let me present you Matteo Ferrara. He is an associate professor of Institute for Cognitive Neuroscience, a lecturer of our master's program, and one of the best researchers in transcranial magnetic stimulation study. Today he will give a very interesting lecture for us. So, Matteo, please.
Thank you very much for such invitation. So today I'm going to talk about an interesting topic, I would say nowadays, which is called neuroenhancement. So we are living in an era where everything is smart. So we have our smartphone, everything is based on smart application. And it seems that publicly there is a need somehow, and I can feel that in everyday life. And I can hear from people, people try to improve themselves, try to improve their brain skills, their cognitive skills. It seems that there is a need to become smarter and smarter. And my research focus is on motor control, processing, and on memory. So I've been carrying out some studies on memory, and especially I'm investigating these processes by using non-invasive brain stimulation. And non-invasive brain stimulation, so by stimulating the brain, it seems that it is possible to increase brain activity. Therefore, it seems that it's possible to induce this neuroenhancement, so to improve the performance of a human being. So what is neuroenhancement basically? So in general words, it refers to this improvement, okay, to this increase of performance, which can range for perceptual skills. You might increase your perceptual skill, motor skills, cognitive skills, social abilities, okay? An improvement which rely on the activity of the brain, okay? But also represents the ability when we talk about brain to facilitate, the different state transition within and between different brain network. So there is not just one specific area which is important, for example, for memory performance. There are different brain regions that they form a network, as well as for attentional processing. There are different brain regions that form a network. If you imagine yourself performing either an attentional task or a memory task, or to do that together, because as I said, the world is running very fast, and we always find ourselves, under the pressure to perform this, to be multitasking, okay, to do more than one activity in parallel, okay? And when we're talking about neuroenhancement, it refers also to the ability, to shift and to facilitate transition within and between these brain regions, brain network, and so also consequently brain processing, okay? And in terms of brain stimulation, and this is our topic of today, when we're talking about neuroenhancement, we refer to the increase of this cognitive performance, cognitive skills, by using some external tools, okay, which can be the use of psychotropic drugs, which can stimulate your brain and can allow you to outperform on a specific task, or by using brain stimulation method, okay? For example, such as transcranial magnetic stimulation. Now, an interesting question is, there is a fairy tale, which, when I was younger, I tend to believe that we are not using 100% of our brain. We are using just 10% of our brain or 20% of our brain, okay? And that is totally false. We are using 100% of our brain. So there is not hidden locus in the brain that are not active and that they need to be activated, okay? The brain is always active. These neurons are always activated. They behave differently between the state of sleep and the state of awakeness, for example. But everything is always active. So we are always using 100% of our brain. If we talk about the percentage, of the anatomical space, the activity of the brain. What does it change actually is how we use our brain and how we optimize our brain resources. As I said, in order to stimulate the brain and to induce this kind of enhancement effect, we can use brain stimulation, okay, which is based on neurophysiology of the brain activity, or psychotropic drugs, okay, by the means of pharmacological approach. But we will focus on brain stimulation today. So when we are talking about brain stimulation, there are two different, categories of brain stimulation. One is the so-called invasive brain stimulation, and one is non-invasive brain stimulation. And the one that we will talk about in this lecture is exactly this, the use of non-invasive brain stimulation. So invasive brain stimulation, basically, the most widely used is called deep brain stimulation, or DBS, and consists in an intracranial electrode, which is implanted in the skull of the patient, and usually on the thalamus, because this technique is very widely used for treatment of symptoms of Parkinson's disease. Once the electrode is implanted, the patient is, through a surgical procedure still, is installed by a remote control under the skin of at the location of the collar here, which can regulate the frequency of the stimulation. This technique allows, for example, in Parkinson's disease, depending by the frequency and adjusting the frequency of stimulation to reduce the symptoms, okay? But it's not, in US, it's been approved, for example, implanted electrode on prefrontal cortex for treatment of drug-resistant depression, okay? But it's not a technique that nowadays is used to induce this enhancement effect, okay, this boost of performance. What instead you can find in scientific literature is the use of non-invasive brain stimulation as a technique to induce this enhancement phenomena. The mostly widely used non-invasive brain stimulation is transcranial magnetic stimulation, or TMS, and transcranial electrical stimulation, or TES. You can see here. And these two techniques absolutely are non-invasive, okay? So the TMS is a coil which is placed on the, it consists mainly by the use of electrical machine which deliver an electrical field through a coil which is placed on the scalp of the subject. The electrical stimulation consists in the placement of two electrodes on the scalp of the subject, and from which then you can deliver electrical stimulation, okay? So what does brain stimulation induce, okay? It induces what is the so-called plasticity change, okay? Better to say that induce not long-term plasticity change, because the effects are transitory, they tend to fade out, but induce short-term synaptic plasticity change, okay? So short-term plasticity change, which can last from 1 day, or in case of the patient, if we are using brain stimulation as a rehabilitation process, after many sessions during the week, you can induce even 1 month, 2 month changes, okay, in synaptic plasticity. These changes, facilitate this enhancement, okay, phenomenon. And also in the case of patients with lesions, like a stroke patients, for example, it helps to recover, and it boosts the recovery of the patient by avoiding the so-called acute state of maladaptive plasticity. That by using brain stimulation, we can drive, I will call it like a good plasticity effect, okay? So, and plasticity refers to the change in how single neurons connect to each other and reinforce to each other, okay? And we can also tell that neuroplasticity refers to the potential that the brain has, okay, to create a new neural pathway, to adapt, to reorganize as it needs, okay? So let's start to focus on TMS, transcranial magnetic stimulation. How does it work? So basically, you can see this is a coil from which the stimulation is delivered. And it was invented in the 80s by a British engineer named Barker, okay? So basically, it's this device that you can see here in the slide. I hope you can see the pointer too. And it's a machine that generates a high amount of electricity, high voltage electrical current. This current goes through this cable until it reaches a coil, okay? On this coil, because of a Faraday principle, a very high electrical fields generate a magnetic fields, okay? The magnetic field is discharged over the scalp of the subject, and it's called non-invasive also because it's not painful, okay? So it's really, it's absolutely not painful. The subject might feel it, but it's because it's of magnetic properties of the field that is discharged, the magnetic fields pass the scalp non-invasively, okay, very easily. Imagine if you have a high electrical current, the skull, the bone of the scalp, the skull, will apply that certain amount of resistance, okay? And in that case, you will induce electroconvulsive shock, which is not really pleasant. But instead, brain stimulation is quite powerful stimulation, and because of the magnetic field properties, can pass the skull non-invasively. Once they reach the first part of the cortex, the magnetic fields, because of the same principle, it encounter an electrical field, which is the one of our brain. And because of that, it transforms itself in electrical fields again, and it induces a local depolarization, okay? The neurons under the coil, they fire, they produce an action potential. I think it's important that you know these notions. So, therefore, if you use a coil with TMS of this kind of shape, which is called figure-8 coil, the maximum electrical field, and so also magnetic field, is elicited at the center of the coil. And this kind of stimulation is quite focused stimulation on the scalp. It has a resolution of 1 centimeter square. According to where you move the coil along the scalp, you might stimulate different brain regions. If you use a single pulse TMS, you can stimulate, different brain areas, okay, with quite high amount of spatial resolution. Therefore, brain stimulation is very interesting technique, okay? And at the very beginning, starting from the 80s, it was used, to study change in behavior, meaning by modulating brain activity, we could observe changes in behavior. And actually, since it's quite focused, and so you can target different brain regions, you can also create a functional mapping of the cortex, okay? You can causally address the relationship between a specific brain region, if you stimulate prefrontal cortex, right, instead of parietal, for example, you can causally address the relationship between a specific brain region and the function of this region. So, if you hypothesize that prefrontal cortex is involved in memory, because an fMRI study showed that, a memory task activates deeper structure like hippocampus, also superficial structure like dorsolateral prefrontal cortex. If you target with brain stimulation the dorsolateral prefrontal cortex, you might induce an interference, okay? So in the memory task. So the performance goes down, okay? So depending also by how many pulses you deliver and the frequency you deliver the certain amount of pulses during a task, you induce an inhibition, okay? So you inhibit the cortex. Well, if you target, for example, the visual cortex, which is posteriorly here, during a memory task which involves the subject, to read a list of words and memorize them, you don't find any change in the performance of the subject. If you target the prefrontal, you will find some changes in the performance of the subject. This is an amazing tool because it gives you like a functional perspective of how the brain works in certain circumstances. You can observe directly the performance going down or going up, we will see later, because we can also use to facilitate, okay? I hope that this is clear. This is a classic setup. The subject perform a task on a computer and receive brain stimulation, receive TMS pulses, okay? We use neuro navigation system to be sure about the area that we are going to target. So first we send the subject to do a structural scan in MRI. We take the MRI scan of the subject, then we use a navigation system live to target specific brain region, in a specific moment of the task. So, today we will focus on a series of studies where I show that there is this enhancement effect. We can induce this enhancement by targeting the motor cortex, okay? This is the motor cortex. This is the homunculus of the motor cortex, all right? So basically, our body parts are represented in the precentral gyrus, okay? So the face muscles, the hand muscles, the leg muscles, along all the precentral gyrus, which represent the primary motor cortex. And this is a very interesting area to see and to observe the effect of transcranial magnetic stimulation. So, for example, because the cortical pathway of motor control goes from deeper structure like basal ganglia, primary motor cortex, and all the pyramidal system, at the level of the cortical spinal pathway, the innervation of the muscle change direction and goes contralateral. Therefore, if I stimulate the motor cortex on the left motor cortex, I can record a motor response on the contralateral hand. I can show you a video on the clip in our lab. It's very interesting for those who are not familiar with brain stimulation. So you can see that stimulation of the motor cortex induce a motor twitch on the contralateral hand. And we can record with some surface electrodes. Here is the navigation system. Here is stimulation of motor cortex. Here is the muscle that twitch. And this is the motor response that we can record, a so-called motor evoked potential, okay? This gives us like a very clear perspective of how TMS, transcranial magnetic stimulation, works, all right? So So how we can, you heard these single pulses, okay? This coil click. This click represents a pulse that is going to stimulate the brain. There are a series of protocols which induce, depend accordingly to the brain regions that you're going to stimulate, inhibition or excitation of that brain region. Therefore, if you inhibit the brain region, it means that usually you use an inhibitory protocol. So every pulse that you deliver, you can deliver at a different frequency. If you deliver 1 pulse per second, you induce, it's called repetitive transcranial stimulation, repetitive TMS, you induce in an inhibitory effect, okay? If you go up from 5 Hertz to 10 Hertz and 20 Hertz, the stimulation usually induces an excitatory event. So the neurons fire. The neurons fire, but after consecutive pulses, there is a cumulative effect that, you know, hyperactivate the area that you are stimulating in that moment. Okay. Then there are also some advanced protocol with repetitive TMS with very high frequency, and it's been shown, like for example, continuous theta burst, TBS, it's been shown that if you deliver 50 Hertz train of 3 pulses every 200 milliseconds at 5 Hertz, so very high frequency, which, you know, repeat every train at 5 Hertz, you induce a very strong inhibition. If you use same protocol, but with some time interval in between, it's called intermittent theta burst, you induce facilitatory effect. Therefore, for example, if you want to show, if you are doing a visual task and you want to improve the performance of your subject, you can target the primary visual cortex of the subject, and you can use intermittent theta burst to induce this facilitatory effect. And so therefore you will induce this neuroenhancement, okay, this enhancement effect, right? So in general, as I mentioned, this is something that, you know, came out during the last 10 years, because before brain stimulation was used to induce especially inhibitory effect, because it would allow to decrease the performance of a subject by stimulating a specific portion of the brain. You might understand which area is responsible for what, not just by passively record EEG, electroencephalography, or functional magnetic resonance to see which area is activated. Because with fMRI, a passive method, you can see which area is activated for a specific task. But to reach that goal, you need to do a lot of analysis in terms of different kind of strategy for the analysis, like subtracting activity from the baseline respect to when the task is on. And that, what you see actually, the activated area that you see, do not necessarily mean that they're so crucial for the task. They might give support to the process, but you don't understand in which extent that area is important for the task and for the process and for the performance of the subject. If you use brain stimulation instead, you target this, you know, networking of brain regions specific for a process like memory, perception, tactile processing, you can observe how much is the impact, okay, of that area on the task. Okay. Please remember that some people, some researcher, they call this as a virtual lesion, okay? But it's not a lesion. You don't deactivate a brain region when you create inhibition, okay? You just decrease the neuronal activation, okay? But you can also, recently, people started to be interested instead, why don't we use brain stimulation instead that, you know, just to map the cortex by inducing inhibitory phenomenon? Why don't we use it for, you know, increase the performance, increase the cognitive skills of the subject? and therefore also to be applied on patients with neurological disorder, patients with lesions, patients with stroke, patients that have stroke and experience aphasia, so difficulties in language in production, and so on. Another technique, which is started to be widely used most recently in a different kind of approach, because 100 years ago, electrical stimulation, as I mentioned before, also electroconvulsive shock was used, but was just delivered by pulses, electrical pulses. But recently, it's been shown that if you apply like a very weak current in the brain, by applying some electrode on the scalp, you can modulate brain activity and you can observe consequences in terms of human behavior. If a subject do a memory task, likewise with TMS, basically. This is called transcranial electrical stimulation, still non-invasive brain stimulation technique. Now, if you deliver electrical stimulation, in brain research, the most common shape of the electrical current that you apply are three, okay? The most used. One is called transcranial direct current stimulation, which is basically continuous electrical stimulation. One is called random noise stimulation, which is, alternating stochastic, random, at different frequency electrical stimulation. It's totally stochastic, okay? It's totally random. And one is called alternating current stimulation, which you can deliver at specific frequency, and is totally sinusoidal, okay? Which attract very much my interest and the interest of many researchers, because the oscillatory activity of our brain works sinusoidally, okay? That's why we have oscillatory endogenous rhythm, like alpha, beta, theta, gamma, okay? So this is what we can record also with electroencephalography. Our endogenous oscillatory activity is sinusoidal, okay? It's not direct. So that's interesting, and I'll tell you just, very soon why. So TDCS, direct current stimulation, is widely used, okay, nowadays. TACS is more novel approach, TRNS as well as more novel approach. So for a few years, people, were preferred to use direct current stimulation because it had some kind of very robust effect, very interesting effect. So in general, for example, this is TDCS, but could be also TACS. This is an example. The two approach, TMS and transcranial magnetic stimulation, transcranial electrical stimulation, are very effective. TMS is more focused. As I said, you have a resolution of 1 centimeter square. TDCS is less focused. You usually use like big electrode. Although I would say that most recently, finally, advanced in technologies allow us to give more focus electrical stimulation by using smaller electrode in specific montage, which is called high definition TDCS or high definition TESS, okay, transcranial electrical stimulation. TDCS or TESS, it's basically, you can see, it's not like biggest TMS, it's very portable, it's very small, it's basically an electrical stimulator with some portable batteries, which allow to deliver electrical current. This is an example of the commonly used electrode to be applied on the scalp, okay? That's the advantage. Maybe it's less focused, but it's also portable, okay? And how does it work, for example, TDCS, direct current stimulation? So basically, if TMS, as you remember at the first slides, induce a local depolarization when the pulse arrives on the cortex. So if neurons are firing, by using TDCS, neurons are not firing. It's not properly a neurostimulation, it's a neuromodulatory technique. So which means that, I will try to explain it like in very simple words, that you might change by using TDCS, the ions concentration at the level of the neuronal membrane, okay? The membrane of the neurons. This ions change affects the likelihood, so the probability that the neuron will fire or not. So the neurons, you know, usually are 65, 60 to 70, okay, microvolts. When you reach this threshold, it's likely to fire. But if you use the DCS, you can increase or decrease. So you can shift the threshold where the neurons is going to fire. By playing with this, you can induce facilitatory or inhibitory effect, okay, by depolarizing or polarizing the neurons at rest. Okay, I hope it's quite clear. An example of a study that we carried on is this, is that we place our subject at rest, we stimulate the motor cortex with TDCS. TDCS can be delivered anodally, you know, anodal TDCS. Cathodal is inhibitory TDCS, because continuous current, direct current, if you see also me, continuous current has two polarity, K minus and plus. Indeed, you have two channels in TDCS. You see the red one here, the blue one here. The red one is delivering anodal TDCS. The blue one is delivering cathodal TDCS. Cathodal TDCS induce hyperpolarization, which means induce inhibitory effect of the neurons. The neurons are less likely to fire after the subject is stimulated with this. That's why if you do TDCS in a task, before the subject perform a task, if you stimulate specific brain region, you will see that afterwards, when the subject is performing the task, you will experience an inhibitory effect. Anodal, instead, is excitatory stimulation, okay? It might facilitate the task. It's inducing depolarization. So, for example, in this study, we deliver TDCS, anodal, cathodal, and sham, which is a placebo TDCS, for 10 minutes, for 15 minutes. Then we switched off the TDCS, and we started to measure motor evoked potential by using TMS instead. You remember this muscle twitch that I showed you? And so in different days, so the same subject in different days. When the subject received anodal, you can observe here an increase of motor response, a boost of motor response. When the subject received cathodal, you can see a depression, okay, a decrease of motor response. When the subject received placebo, you know, basically the motor response does not change significantly. This is effects of TDCS. It's an offline technique. So you apply TDCS, and then after you switched off, this gray bar, more dark gray, is time where the subject is stimulated. And there are no effect, more or less. There's just a tiny effect here, but they're not, in the normalized data, there are no significant effect. And when you switched off TDCS after 10 minutes, 15 minutes stimulation, which is the standard that is usually used, you start to see the facilitatory, the boost, or inhibitory effect, according to the direction of the current. What are we focusing today instead, in terms of neuroenhancement, is the use of alternating current stimulation, okay? So why? It's very interesting, because since our brain, you know, elicit oscillations, okay? Oscillatory activity. When you sleep, you have slow waves. When you are awake, you have high frequency wave, alpha, beta, and so on. So the idea was, if we deliver alternating current stimulation from the external by using electrical stimulation externally, and we try to interact instead just to inhibit and induce purely inhibitory or excitatory phenomena, we try to interact with the endogenous oscillatory activity, our oscillatory activity. Can we somehow modulate brain activity? Can we boost, for example, brain activity? And one isolated study at that time of 2006 that I found is is a study published on Nature by Lisa Marshall, which she delivered basically. She called a certain amount of subject, and she asked the subject to memorize a list of words. So the subject arrived, the subject is receiving a list of words, the subject needs to learn these words, and then the subject goes to sleep. And she was recording EEG during sleep. While, in sleep is divided in different stages, stage 1, stage 2, stage 3, REM sleep, etc., etc. Okay? While the subject was in deep sleep, she was delivering alternating current stimulation at very low frequency, because this is low wave, lower than delta waves. And some subjects receive stimulation, some subjects, they receive placebo stimulation, sham is called. And amazingly, what she discovers is that the day after the subject wake up, they were asked to, do you remember the words that you read yesterday? And she discovered that those subjects who received this sinusoidal stimulation, alternating current stimulation, were able to recall more words. Okay, so she boosted the memory. So she induced a neuroenhancement effect, okay, on memory performance. And she used a 0.45 hertz, but she used also 2 hertz, if I'm not wrong, another frequency as a control. So only that frequency, 0.45 hertz, was inducing the effect, okay? So meaning that the stimulation, the frequency while you're delivering a sinusoidal stimulation is very important, induce frequency-specific effect. Then after a few years, since when I was working in London, we were fascinated by the use of alternating current stimulation, while all other people were using TDCS, direct current stimulation, we decided to stimulate the somatosensory cortex of the subject, which is important for tactile, processing. When you grasp, when you recognize with your finger, different texture, all the somatotopic processing, and the proprioception, processing. And we delivered in our group of subjects a very simple study. We applied the stimulation on the somatosensory cortex. We delivered different frequency of alternating current stimulation. And we discovered that while the subject was receiving stimulation in alpha and especially in gamma, also in beta, but lower gamma, mid gamma, theta, delta, no effect. The subject were experiencing kind of tactile feeling, okay? So again, we induce a frequency-dependent effect of TACS. And what is interesting is that studies with EEG electroencephalography on tactile task, like for example, grasping an object, they show exactly that the frequency of the frequency of the oscillatory activity of the somatosensory cortex is prominent in alpha and in gamma. That means that likely with our alpha and gamma stimulation, we entrain the endogenous activity. We got face locked with endogenous activity. But this was not yet proof. That was just our speculation. So same story on the visual cortex. People were experiencing phosphins, visual flickering in another study by a colleague, when they were receiving 20 hertz stimulation rather than 5 hertz stimulation or 40 hertz stimulation, by stimulating the visual cortex. In EEG 1020 system is OZ, okay? Respect to frontal cortexes. But it's not so much important, so it's important that you get the point. The point is that different brain regions responsible for different brain processing, like visual system, somatosensory system, prefrontal and parietal for sleep, in the case of Lisa Marshall for long-term memory, and different frequency. The frequency are different, but because these brain regions, they communicate at different frequency, okay? So we have a specificity. So the idea that we had is also to study on the motor cortex different effect of the frequency of stimulation. And we ask, we place an electrode here on the motor cortex to do alternating current stimulation. We use TMS, the previous technique that I showed you, just as a probe, as a test, just to deliver single pulse to, record motor activity from the hand. And we ask our subject that they were at rest, not moving at all, in one condition to stay just resting, okay? And in one condition to imagine a movement. Can you see? I hope you can see myself. Imagine this movement, like this pinch to grip, okay, movement, without to move, just to imagine that. Subject were eye closed, there was a warning, and the subject has in one condition just to stay at rest, and in condition to stay at rest, but imagine, use imagination of this movement, okay? Imagination of the movement always activate the motor cortex and increase the motor evoked potential. What we did, we had a baseline, no electrical stimulation, then we deliver 5 Hertz, 10 Hertz, 20, 40 Hertz. We stimulate a side control at 20 Hertz and another baseline, okay? And what we saw is that during the stimulation, of course, in general, motor imagery increase. You can see on the panel A, the motor activity, okay? Boosts, activate the motor cortex. But if you subtract the classic differences between imagination and rest, and you check the effect of alternating current stimulation, we saw that for motor imagery, theta and alpha, in a lesser extent, where, theta and alpha here, boost motor imagery drastically. Look at this difference and at this difference. It's really huge. While at rest, only beta increased the cortical spinal output. So again, we had this frequency dependent effect of TSS. So that was really cool because motor cortex, when it's at rest, if you record EEG, is at 20 Hertz. We give 20 Hertz, we increase the motor activity. If we deliver 40 Hertz stimulation, nothing happens. When you imagine a movement, you have activation, you have modulation of alpha activity. You can record it by EEG. And when we deliver alpha externally, we had a boost of motor imagery, which means that when the stimulation arrives, if the endogenous activity is nearby the same frequency, it got phase locked and it boosts. Okay, create this boosting effect. Now, since we were very excited about the imagination of the movement, we decided to investigate what's going on when instead we want to check the mirror neurons effect. It's very well known that there are some neurons in our sensory motor system that respond when we observe a movement. For example, if now I'm doing this with my hands and you're watching me, like I'm doing this like I'm a dumb person, actually your sensory motor system activate. Okay, there are some categories of neurons which are called mirror neurons, which respond to movement, and they respond not only when you observe the movement, but also when you perform the movement. So in the monkeys, we show that the monkey grasps the food, mirror neurons respond. If the monkey see the experimenter grasping food, the same neurons respond, okay? They're called mirror, okay? It's very interesting effect. Indeed, in our lab, if we do TMS, while the subject is watching a steady hand, observing a steady hand of the other person, or while the subject is not imaging, this has nothing to do with the previous motor imagery. While the subject is just observing a hand movement, you can see the motor evoked potential that you record. The subject is at rest, no moving, nothing changed in his physical state except brain activity, okay? So the sensory motor system activate. Therefore, when they receive the pulse of TMS, the motor response is higher, okay? This is a classic mirror neurons effect, which you can measure by TMS. So to complicate our life, we decided to use a mirror neurons task to record from the muscle that you saw before in the movie clip, which is FDI muscle here, which is the muscle where you perform the pinch to grip, and the ADM muscle. A muscle is not involved in this movement that the subject observes. This is a better picture, okay? So the actor is here with the hand. The subject is here resting, and we are recording muscle activity. The subject has both the electrode for alternating current stimulation, because we wanted to investigate the effect of boosting the mirror effect, and the TMS to check the motor response. And then this is another perspective. You can see that the actor is either conditions, showing a steady hand, and in another situation is performing a movement. The subject is just observing this movement. And what we showed is that AO represents action observation, the subject observing the movement. Rest represents rest, subject observing the steady hand, especially in the muscle, which is the prime mover of the mirror neurons. We had a very strong boost when the stimulation was in alpha and when the stimulation was in gamma. So alpha and gamma alternating current stimulation significantly boost the mirror neurons effect, okay, which is another process, the motor imagery. So we can use this kind of technique to boost performance and to boost brain activity. Indeed, this is sham, placebo, no electrical stimulation, just TMS to check motor response. This is the difference between rest and action observation. This is the classic mirror neurons effect. But look at this graph, how much this boost goes up when the subject receives alternating current stimulation. Some effect we saw also on the other muscle, when stimulation was, delivering gamma band. Okay, that's very interesting. Indeed, basically this is a summarizes these results. When you observe a movement, alpha and gamma boost mirror neurons effect. When you are at rest, only beta boost the motor activity. So there is a frequency specificity. If you change your mind state, the oscillatory activity of your brain change. And the frequency of stimulation that you receive, modulate differently according to your brain state, which in somehow gives us a very cool insight about which frequency is responsible for what. And then now that I know that, I give you externally this frequency and I boost this process. Another interesting study that we ran recently and we published in this year with my former master student in our lab is that it was to check what's changed when the stimulation, alternating current stimulation, is delivered offline before, like TDCS, 10 minutes, then you stop it and you check the effect. You see, this is a time course. First, we give TMS, we record motor evoked potential, you see? Then we apply the stimulation, we don't touch the subject, just we apply electrical stimulations, alternating current stimulation. And then, again, we measure motor evoked potential for a certain amount of time to have a time course of the effect. And the second one is the online experiment. We check motor evoked potential before the electrical stimulation, and then we measure the effect of TACS during, by using TMS online. So while the stimulation is ongoing, the electrical, we give also the magnetic over. Okay, then we take a break, again. So, because what is important question is this: what's happening when I'm stimulating the brain? Is the effect, this boost, happening while in the moment that I'm stimulating the brain, or with some different technique like TDCS or high frequency protocol TMS, or this effect happens after the stimulation is off? So you condition the area with stimulation, and then after you have this after effect. In the case of TACS, we discovered that offline TACS does not induce any effect. we deliver alpha, beta, and placebo. And then we measure motor evoked potential after the stimulation. And you see there are no differences. But when we do online, so when we measure motor evoked potential during online, we found again that subjects at rest, have a boost of cortical activity when the stimulation is delivered in beta frequency at 20 Hertz. Okay? I know that might sound everything so much complicated, but in reality, everything is very easy. Brain oscillates at different frequencies. According to the frequency you're delivering the stimulation, you can boost brain activity if that frequency of stimulation is near, it's really similar to the frequency that in that brain region, is generating. Okay? Then, in my former lab, again, we wanted to use alternating current stimulation. How strong is this effect? To boost fluid intelligence, okay? In an intelligence task, we moved from motor, and then we say, okay, let's see if this effect is also strong in cognitive task. And then we stimulate the middle frontal gyrus, which is usually involved in tasks like Raven matrices, which is a classic intelligence task, which there are different ones, and there are 3 difficulties from relational matrices, and one is the logic matrices, okay? And what we saw is that for the logic matrices, delivering alternating current stimulation at theta, alpha, beta, and gamma actually boost the speed of performance only for logic matrices. Okay, which actually they adopt a completely different strategy in general respect to the relation matrices. So gamma stimulation on middle frontal gyrus boost while you're receiving stimulation, because I just show you that the effect is during the stimulation, not afterwards, doesn't last with alternating current stimulation. That's the issue, doesn't last. It takes place in the moment you are receiving stimulation. You can boost the speed of performance of fluid intelligence, meaning according to the logic task. That was really interesting study. Then, again, the frequency-dependent effect of TSES, you see, because only gamma, we use different frequency, and only gamma does this. So then the same, this is not a study where I participated, but it's a follow-up of the previous study where I participated. Then in Oxford, they checked, and this is an important point, guys, is that the effect of brain stimulation, of this boost of performance, has been investigated in this sense. In this study, they repeated exactly the logic matrices and the relational matrices as well. They use also a working memory task as a control. But what you need to know is this, the task is this, okay? But the difference is that they divided 2 groups of subjects. Those who, before to start the task, they did a questionnaire, a classic IQ test, and those who have in 2 groups. They recruit a subject according to those who have high IQ and low IQ at the test. What is interesting is that the effect of gamma stimulation is high only for the, this is the reduction here that you see means the faster response time, only for those who have a slow IQ before the task, okay, at the test. Those who have high Q, they don't benefit from brain stimulation. And that's exactly the point. What this neuroenhancement and people want to increase and become smarter and smarter, it seems instead that the brain stimulation is inducing this enhancement effect because it's helping to optimize the cognitive resources, okay? To boost the cognitive activity, the electrical activity of the brain, in people that are already high performant does not change so much. The effect is almost null. Those who instead who are low performance at the beginning, they benefit, slow IQ guys, they benefit much more of the brain stimulation. I hope the message is clear. Now, another interesting study, how much in which interesting way we can boost brain activity this was published in Nature Neuroscience from a German group, is that alternating current stimulation can induce and can boost lucid dream. Okay, what is a lucid dream? It's a consciousness phenomenon inside the sleep, when you are dreaming and you are conscious inside the dream. Okay, not these guys, they did not just recruit a group of subjects and apply stimulation and they induce lucid dreams. Okay, they select a subject that there are specific questionnaire and also hypnosis questionnaire. There are some subjects, some people among us that are more susceptible to experience lucid dreams. Okay, so there are specific questionnaire you can select subject, you know, that they usually have this kind of phenomenon. Then once they screen out the subject, they recorded EEG during sleep. You remember the very first study, okay? While recording EEG during sleep, they were delivering alternating current stimulation at different frequencies on these subjects that are susceptible to lucid dream, but also on those who are not susceptible to lucid dreams. They had two groups. And what they found is that when the subject received 25 Hertz in gamma frequency or 40 Hertz still in high gamma frequency, respect to very high gamma, alpha, beta, theta, sham, placebo, whatever, only for these frequencies, at these frequencies, the subject has a boost of this consciousness inside the dream. Okay, so basically, how does it work, alternating current stimulation? This is a very cool study. Okay, so subject, after they wake up and they have specific tasks, they have to describe the dream and they experience this, is called insight. There are different parameters. Okay, control of your dreams, control of the consciousness inside a dream, and dissociation. Okay, and by using this frequency, in the case of control only 25 Hertz, subject boost this kind of phenomenon. You know, they had a boost, they had an improvement, they had an enhancement of this consciousness inside the dream. So how does it work, alternating current stimulation? The idea is that you record EEG and you see that our brain oscillatory activity change. You know, there are different rhythms, alpha, beta, theta, etc., etc. When you give TACS, alternating current stimulation, in a specific brain region, you actually, if you deliver alpha, and this rhythm is in alpha range, you actually induce this entrainment phenomenon. You interact with endogenous activity, you anchor, you get face lock, you see here, and you boost it, as long as the stimulation is ongoing, okay? So, alternating current stimulation induces frequency-dependent effect. Specific frequency on specific processes induce a boost, okay? These effects are state-dependent. You remember motor imagery. If I change mind state or action observation, if I change mind state and I'm not doing an imagination task, the frequency that before was boosting now is not effective on myself. It's another frequency, because when you change mind state, you change your endogenous oscillatory activity, okay? And these TACS induce entrainment effect. It interacts with endogenous activity, okay? These effects, this is very important, are individual performance-based, okay? As you remember, the study on fluid intelligence. If you are low performance in an intelligence task, you had benefit of stimulation. But if you are already high performer, you don't become a Superman. Okay? You have a ceiling effect. Are TACS effects robust? Well, I wanted to investigate, you know, if it's possible to modulate the shorter memory capacity, because it's a very stable process. So, you know, if you need to memorize a phone number, you use your memory span. If you need to count and remember the number of the items in your basket, if you need to enter a pin to go on. So the classic span memory task is the digit span. For example, if I tell you a series of numbers, 3, 2, 7, 1, you need to repeat in the order. Okay, if you make a mistake, you have another chance, and then, you know, after 2 consecutive mistakes, that is your span. But if you are good and you repeat correctly, I can add another digit until I don't understand which is your memory span. 7 items, 8 items, for example, I can ask you, I can tell you like 6, 2, 5, 1, 4, 3, 6, 4, 3, 8, and then you have to repeat immediately. Okay? If you repeat correctly, you go on. If you commit 2 consecutive mistakes, your span. In general, from 20 to 40 years old and high level of scholarity, the span forward is around 7 items. Okay, 7 plus 1 or 2 standard deviation. Okay, but then there is also the digit backward. Okay, so I can tell you, for example, 1, 5, 2, 6, and you need to repeat backward. You need to do a mental rotation, 6, 2, 5, 1. So I decided to stimulate the parietal cortex, which is involved in this memory span, and I deliver different frequencies. And I decided to use 2 groups of subjects, young subject and middle-aged subject, because it sounds very scary, guys, but cognitive decline by recent study has been shown that start from 35 years old. Okay, that's why usually people, when they study young and old people, differences in brain activity, never study what is in the middle. So that was also our purpose. And what we show is that middle age, which were from 35 to 50 years old, stimulation has no effect. So first of all, no results by stimulating the parietal cortex on digit backward. The subject received backward and forward task to perform. But for the forward task, young people, when they receive beta stimulation on the parietal cortex, and the parietal cortex, there are lots of studies that they show the parietal cortex activity in beta is memory related. So therefore, we come back to this idea that we interact with endogenous activity if we deliver the frequency corresponding to the brain region's frequency. And then we have a boost of the span. Okay, sham here is placebo. 6.5 is the average span of the young adult. And look, 7.2, 7.3, we really increase and boost the digit span activity. And you need to understand that why I wanted to run the study is because if your span today is 7, in 1 month it's still 7. So it's a very stable, very robust effect. And if not of the stimulation of the, the memory span is very robust, stable. In 2 months, if I test you again, your span is still 7. But what changed the span is aging, when you're getting old, and scholarity, okay? And usually this changing are around in the order of 0.5 to 0.8 of the classic digit span. So if it's 7 at 25 years old, when you will be 45, or when you will be 55, eventually it can be 6.5. But look how much we boost in 1 day of stimulation. So from 6.5, almost 7.2. The boost, these are all the subjects, all the group together here. The beta stimulation, the boost, correlates with the age. The younger you are, the higher is the boost. Okay, and this is because the brain activity on the parietal cortex, there are lots of studies on that, actually changes in ages. So the beta is not synchronous anymore, and it's been shown already from 40 years old and on that the beta activity desynchronizes and becomes bilateral. So here we stimulated unilateral. A young has synchronous beta activity, and when we deliver synchronous beta stimulation, we had a boost. That's likely why middle age did not benefit of the stimulation. Okay, so we can boost brain activity, but inside certain limits. Okay, so this boost is strong, but it's not like huge that you say, wow, I outperform. Individual differences must be taken into account. Remember the study with intelligence, okay? If I'm a low performance, I benefit. If I'm a high performance, I don't benefit, okay? But also training with brain stimulation actually induce greater neuroenhancement effect. The effect of stimulation per se, it's significant statistically, but it's never such huge that people say, wow, you know, I can boost my brain activity. If you associate training, you have a higher boost. This is a study, I try to explain you very fast and very briefly because time is running out, where the subject has a numerosity task, is a cognitive task, okay? Where the subject has to discriminate numerically more yellow or more blue dots, okay, and give a feedback. Or where the subject has to discriminate continuous quantity, okay? Is this a longer line, space, or time, you know? You can see here. Then they were an arithmetic task and an attentional task. And what they did here, these guys, they used TRNS, random noise stimulation. I don't go into detail now on that, but it's very interesting in terms of the training associated. They did parietal electrical stimulation. There was a condition only training without stimulation, and a condition of motor, stimulating a control area, the motor, which is not cognitive, so it's a kind of control condition, and a condition where they stimulate the parietal again, but without training. What is important is this: stimulation only, training plus stimulation. And what they show is that when the subject, you know, this line down is stimulation plus training. When the subject receives stimulation plus training, the Weber fraction is an index of performance. The lower it is, the higher is the performance. The subject performed greatly better. They had a boost of the discrimination task. Respect to training only, which is this, okay? In respect to stimulation only, which is this. So if you associate training with brain stimulation, you have higher boost. And for time discrimination, same story. You see this black line, pre-training, post-training. If you do parietal stimulation plus training, you have a high significant boost, okay? Also, just before to finish, I can say that another important factor that affects brain stimulation efficacy, so it's not that you can boost every time, it depends, there are individual differences, there are also some genetic factor, is the BDNF, which is a brain-derived neurotrophic factor, okay? So the brain-derived neurotrophic factor actually is a protein that code the homonymous gene BDNF, which it's called promotes neuronal growth and brain plasticity. It promotes development of our central nervous system when we are born. And is also important for brain plasticity. Indeed, those who have a polymorphism has been shown to have, for example, reduced hippocampus volume. And people will think, oh, if my hippocampus is reduced, that means I have lower memory performance. But no, there are some strategies to correct that. Indeed, we ran a study, the VAL66 met is the polymorphism of this BDNF factor, okay? And let's say that 18 to 30% of the population, even among us, there is someone that has this polymorphism. It doesn't mean that it's sick or whatever. It's actually very healthy. It's just that, has a polymorphism which can affect the strategy with which he can, for example, perform cognitive tasks, but still can perform very high, okay? So the meta-allele is associated with variation in cortical morphology, as I said, worse memory performance in some cases, but I can tell you the recent literature, they show that the performance is not much affected because people use other kind of strategy to compensate, and is linked with neurophysiological disorder. So reduced response to non-invasive brain stimulation protocol that alter cortical excitability has been shown. Now, since brain stimulation induces cortical plasticity and BDNF is important for plasticity, why not to see if we can or we cannot modulate brain activity on those subjects who have these polymorphisms, and why this is important. So we target the prefrontal cortex here, which is involved in the memory task. We submitted a long-term memory task to our subject. I'm going to get brief. So basically, classic old new task. Did you see this word before? Yes and no. Yes and no. We were delivering transcranial magnetic stimulation. We had two groups, Val-Val and Val-Met. Val-Met are those who carry this polymorphism. As you can see, the performance between the two groups in general doesn't change so much. But look at the no TMS condition without brain stimulation. You can see that the performance more or less is not drastically different. Of course, deep and shallow are two conditions of the task. Deep is more semantic, shallow is more perceptual. Yeah, Val-Val maybe have higher performance in semantic, but not significant. Statistically significant. What was statistically significant was that when we applied TMS on the left dorsolateral prefrontal cortex during the memory task, we reduced the performance only on those who have not the polymorphisms, which means that who has not the polymorphisms is more susceptible to plastic change because, you know, the BDNF is plasticity related. The TMS induce plasticity change. And indeed, when we spot the right place in the brain, look at that. We have a reduction of the performance. But the Val-Met, those who have a polymorphism, when we target the same brain region, no changes, no effect of TMS. Okay? So many factors affect brain stimulation effect. Okay? Now, the take-home message is that there are lots of companies that use brain stimulation as a, electrical stimulation. This can be used as a TDCS. They sell this product online, okay? And they tell you, oh man, overclock your brain for gaming. There are lots of companies, and buy it for $300. Then you arrive this kind of package, which is compatible with an app, very pretty, very cozy. You're home and say, okay, now I follow the instruction. I put this while I'm reading, maybe I will read better. While I'm running, maybe I will run faster. No, it doesn't work like that. You can see that, first of all, what I show you, the real evidence in science is that you need to know which area to stimulate. Look at this montage of these electrodes. Mostly they target the prefrontal cortex. So if you want to do motor task, there's no way here you can stimulate the motor area. So you need to know the brain regions. You need to know the frequency of the brain regions. You need to know lots of factors that I just showed to you. And this also, overusing of this stuff at home can be also a little bit dangerous. There's no there's no medical doctor that is following you while you're using brain stimulation. If you are a patient, you can use this as a treatment, but we can use better stimulator, not just with this montage. We can place the electrode in the right place and to deliver stimulation at the right moment. Otherwise, guys, you can buy as much as you want this. You just have placebo effect. Otherwise, it doesn't work. Okay? That's the take-home message. So, do you want to boost your brain function? Then forget to become a superhero, okay? Because, as I show you, you have what brain stimulation does, and when you see this boost, is because you are optimizing your brain resources. You are not adding up something, okay? So, brain stimulation per se, do you remember the study about the training together with brain stimulation? But I also studied brain stimulation together with medical drugs. That you have a very big boost. But brain stimulation per se induce, yes, changes in performance, but they are tiny. They are not so huge. Very important changes can be used for rehabilitation process, but for clinical application, neurorehabilitation. But don't expect magic. That's what I'm telling you. Okay, don't buy this stuff. It's a waste of money. So thank you very much. And if you have any question, I'll be glad to answer. This is my group at HEC. It's Memory Motor Control Group. Inside the CDM, which is Center for Cognition and Decision Making at the Higher School of Economics. So we have different groups that perform different kind of research, and now we have an Institute of Neuroscience. We have lots of cool stuff. We do EEG, MEG, magnetoencephalography, functional magnetic resonance, brain stimulation, as you saw already. And this is our master. It's very cool master program, to be honest. A couple of the studies that I show you are from my master student, actually, that we published. Very good. So I'm very happy of the environment and the laboratories that we have, and the master program is very cool. You have different kind of topics, brain-computer interface, signal processing, advanced neuroimaging techniques, cognitive neuroscience, and on and on. So thank you for your attention.
Thank you. We have questions from audience. So the first question, does TMS affect consciousness at the moment of stimulation? Are there any temporal psychosis-like phenomena in its sites affect list?
Okay, so the first question I understood, and I can reply now, which is yes, it can. There are some interesting, at the moment of stimulation, yes, induce it can affect consciousness. And it's kind of covert process, because usually there is a like very top level specialist and scientist, which is called Marcello Massimini, and the other one is Giulio Tononi, which are experts of consciousness, and they run several studies during sleep, and they show consciousness phenomenon in the brain taking place during sleep, and also in patients in coma. So yes, there are people studying this, and it's possible to do this. And the second question is, okay, yeah, side effect of TMS. I have no current knowledge of any psychosis-like effect. What you need, there are some international guidelines of TMS. You cannot deliver more than, I just give you an example, more than 900 pulses per day, more than certain amount of time for a very high frequency of stimulation. What you can induce is an epileptic seizure. So an epileptic crisis, temporary. In a one, it's just transient, and some people might experience, and this is one thing, but never happened because we always use TMS inside the guidelines. And another side effect eventually is that if you stimulate brain regions prefrontal, like here, you are, since, you know, when the magnetic fields cross the scalp, reach the cortex, is electrical stimulation, but here you have also your facial nerves. So if you stimulate here, no problem. If you stimulate in the orbital cortex, since the nerves, transmit electrical information, so we're talking about electricity, you don't stimulate just the cortex, but also your facial nerves. So the subject experiences some muscle twitch in the face. And this can be uncomfortable, and in some cases can be painful. But the subject report, you orient the coil differently, then you're good. Otherwise, you stop the experiment. So the subject has not to feel pain. Otherwise, pain also affects the task, and the effect that you get is because of pain or uncomfortableness. But the main problem with TMS is to avoid to induce epileptic seizure. But this is fine because it's controlled. Other question, I don't know if I find effects of NIBS, like a very heavy side effect of NIBS, are very, very rare, because there are some guidelines on how, for example, in the case of TMS, especially because it's magnetic field and it's quite strong stimulation, because you know the neurons fire, you have a muscle twitch, that means neuron fires. In the case of electrical stimulation, it's so weak stimulation that you don't experience mostly negative side effect. You might feel some, the electrodes get warm on your skin. You might feel some tingling sensation, can be uncomfortable, but nothing serious, even for longer period of stimulation. But there are guidelines for this. You cannot stimulate, for example, with TMS, as I said, more than 900 pulses. What can happen if you insist stimulating a person, which does not happen, for very, very long time? The person can lose consciousness, can faint, because you induce a very, very acute, a very strong epileptic seizure. A negative effect reported and published, like tested and surely negative effect, are also sometimes reported on patients that have a brain lesion, and which they agree instead to have more aggressive treatment out of the guidelines to do recovery. And in that case, it can happen you induce, epileptic seizure. You can control this, and you just follow the guidelines. You can damage a person even just not by using the electrical magnetic fields, just by hitting a person. What I mean is that if you follow the rules, nothing will happen, of course. Very, very unlikely. Very unlikely. Unless, an important thing is you should have metal implants in the head because it's a magnetic field. In that case, you can use big damage eventually if you use strong stimulation. So there are some protocols, there are some consent form, there is an ethical committee, you need to respect that. The subject that arrives has to declare that has not metal implants in the face or in the head.
So one more question. What are the prospects for treating coma?
So if you can induce a person in coma with TMS, never heard about that. Never heard. And I don't think there is any evidence of that. To the best of my knowledge, it's it's absolutely safe. It's non-invasively. It's not painful. And there are lots of guidelines. People used to respect that, of course, because you don't want to damage a patient. So that's why it never happened. You don't kill neurons unless, you just modulate that activity. You don't deactivate a cortex. You don't induce a real lesion. That's why some people call it virtual, okay? Because then it's fade out. It's not that, it's a magic tool, but I just give you an example. When a patient suffers from an acute stroke, the brain tends to repair, okay, to compensate the damage, and sometimes doesn't do a good job. That's why even after stroke, after the acute period, the patient instead to get better, get worse. And this is called maladaptive plasticity. As I mentioned before, TMS is able to induce plastic change, short-term mostly, but also long-term if you have a daily session of TMS, which helps to boost the forming of new pathways. So new fibering, new connection, okay? So you help to boost this kind of, you induce this long-term potentiation, you strengthen the neuron activity, which helps to do instead of a very good neuroplasticity effect, which helps also to boost the neurogenesis in that case. I don't know if I answered the question clearly for the... Hello?
Yes, thank you for your answer.
Yeah.
So now we have to finish our lecture. Thank you all for coming.
Thank you very much, and thank you very much for the invitation. And if you want, you can also write me down or the question. I can also give my contact, email contact, if you people and audience is interested, they have other question. I'm happy to reply to further question. Really, thank you very much.